Hello everybody. I will present today our preliminary results of the study Gender Effects in a Subsample of the Human Connectome cohort. The study is based on simulation, and my background is the research group Neuromodulation and Multimodal Neuroimaging. It's located in the University Hospital LMU in Munich, Germany. We've done in the past a lot of different multimodal neuroimaging studies. Today we will focus only on the modality of T1 weighted and T2 space data from the Human Connectome project dataset. This data is freely available, and we do the simulations exactly as we have done in the past, with 2 milliamperes, and the electrodes will be located on the left DLPSC and the right DLPSC. There is anatomically variability. If you are looking into the cycle deaths, that's a study by Hill and colleagues in the Journal of Neuroscience 2010, then you will see differences that go up to a factor of about 7 in the adult brain, and even in the infant brain, we are here in a factor of about 4. And this variability is even found in monozygotic twins. And when we are looking into one of the largest data sets so far in the Enigma cohort study from Kong and colleagues, then we see that sex is strongly influencing their cortical thickness in areas also including huge areas of the frontal brain, and also the brain size is completely influencing cortical thickness. We are applying on this variable brains non-invasive brain stimulation. Our focus in our study was to investigate gender and brain size effects in regard to simulation on a larger cohort of healthy subjects. The sample we used is based on 450 subjects in the age range of 22 to 36. There are a gender distribution of about 250 females and about 200 males. The structural data has a spatial resolution of 700 micron isotropic T1 weighted and T2 weighted images that were all acquired by the Human Connectome project. There are detailed demographic and behavioral data and also multimodal MRI. There are 210 non-twins, 130 monozygotic twins, and 106 dizygotic twins. We have used Synapse E-field computation using the Salad Atlas ROI and a proxy of it. The Salad ROI is based on frontal cortex atlas estimation by resting state and DTI in humans and macaques. The proxy we have applied is based on the MNI coordinate and a sphere of 18 millimeters. It's shown here in a blue binary coding, and here it's overlaid on the solid areas in this axial slices. The method of Synapse simulation has been published by Windhof, Alexander Opitz, and Axel Keeser in 2013, and it's based on T1 weighted data. You could also include T2 weighted data. They use FSL and FreeServer, and they take into account skin, scalp, eyes, and tissue groups like gray matter, white matter, and CSF. They are all different weighted, and when you do simulations, On the motor cortex, you see that with increased E field, with increased stimulation strength, you get higher E fields. Also, like the stimulation side matters, so meaning if you locate the electrodes more anterior or more posterior, you will have a different distribution of the E field. We have used FreeServer 6.01 with LERM at the LZ Supercomputing Center in Munich. Thereafter, we used the SIMNIPS based on our own NAMNI scripts on a local cluster. The results. We found a remarkable effect of gender on the E fields in the DLPFC region of interest. Their effect sizes are moderate, but the sample is really large with 450 subjects. Their E fields, the mean E fields, are associated with an increased intracranial volume that has been found to be larger in males than in females. We did not find any age-related effects on the DLPFC ROI in this cohort, but this could also be related to the fact that their age range is in a younger age. If you're looking here more specific into the different percentiles calculated by SIMNIPS, you will find a strong effect of the intracranial volume, here shown in the form of different distributions for their gender, and also are quite different E field results based on gender. Here shown for the 95 percentile, 99 percentile, and 99.5 percentile for the norm E fields. There were no gender effects. Here in this picture, it's a different display from the just like distribution plots, and here you also see that for males, it's for all 3 percentiles quite larger. When we go into more robust statistics like Bayesian statistics, we see a strong effect starting with a sample size of about 200. We've done in the past one study that has been published by Lucia Bulabas from our research group, where we looked into different solid left DLPFC regions of interest and used the data of the today largest clinical trial on major depression. There we found that increased gray matter in the left DLPFC is associated with an improved antidepressive response. The interesting fact is that this was only specific, this gray matter area, for the modality of prefrontal TDCS. It was not given for the antidepressive medication or the placebo condition, the two other arms. Their effect sizes are shown above, and these effects do reflect some preliminary interpretation that we could use DLPFC gray matter for therapy planning. Why this is relevant for psychiatry? If you're looking into this large meta-analysis of 193 voxel-based morphometry studies, it clearly shows up that the bilateral insular cortex and the ACC is affected across different psychiatric disorders such as major depression, schizophrenia, or bipolar disorder. This has also been found for the resting state data. This study has been replicated by Shah and colleagues in biological psychiatry, and they again found these areas. They are also, for the resting state, are known as the salience network. Interesting. When we are looking back to our studies, starting with our frontal stimulation paper on resting state, from 2011, we found some medial areas where we found the strongest significant effects. You find those effects also in the simulations between those electrodes, but also in the effect for the task-based fMRI and even in the EEG. Here is some meta-analysis on new query on major depression. And again, when you include 896 studies, you find a pattern in the left DLPFC and the ACC that is strongly affected by the disease. We think that with the stimulation, we are on the right location. We found in our study here a significant gender effect on E-field strengths. We found no effect of age in healthy subjects, but we've done previously a study by Mitsutani and Takahashi that is in preparation, where we found that for diseases like major depression, schizophrenia, there is an age effect. So meaning a decline in E-fields depending on their age. That has also there not found for their healthy subjects. Their ICV, the intracranial volume or the brain size, is directly associated with E-field strengths. On this data set, it will be interesting to look into genetics because we have in this cohort monozygotic twins, dizygotic twins. And another interesting factor will be the layer fMRI, as the human connectome cohort also includes 7 Tesla data. The question that remains from this study is, should we use individual-based simulations before we plan our next studies or before we apply stimulation to patients? Thank you very much for organizing this exciting conference. Also, many thanks to all the people contributing to the study. The most of them are also part of the research group Neuromodulation and Multimodal Neuroimaging. I'm open for the discussion and will just like announce that we will also publish all our scripts on Zenodo. Thank you.